What's happening, guys? Arush here, and today we're talking about chromosomes, and we're going into an introduction into the cell cycle. So this is actually two different concepts today, but first we have to go over what chromosomes are. Okay, so let's look into this. If I look at this picture, you could see that this right here is my cell, here's my nucleus, and if I were to zoom in into the center of that nucleus, all that stuff that's inside, I would see these strands, and it looks like these strands that have little beads on them, that's actually our DNA. And those little beads, those are called histone proteins. What's going on is that's our DNA loosely wrapped around these histone proteins, and it kind of helps to keep our DNA organized, okay? When our DNA is in this state of being loosely wrapped, we call that chromatin. So anytime I say that word chromatin, I'm referring to DNA that's loosely wrapped around these, loosely wrapped around these histone proteins, okay? And I refer to that as the spaghetti state, okay? It looks like a bowl of spaghetti. There's our DNA loosely wrapped. It's coiled around these histone proteins that kind of help keep it organized, okay? But now what about when our DNA is tightly condensed or tightly coiled? Well, now we have what's called a chromosome. So if I take a look at this strand of DNA, it starts to coil around the histones, okay? Those histones begin to coil around themselves, and it keeps getting tighter and tighter and tighter until it forms this structure right here, this X-like structure, and we call that a chromosome. So essentially, a chromosome and chromatin is the exact same thing, it's just the different state that they're in. If it's loosely coiled, it's chromatin. If it's tightly coiled and in this X structure, that's a chromosome. Okay. Now, here's one thing to keep in mind too. This part could get a little bit tricky, so bear with me. This chromosome right here, this is one chromosome, but it has a duplicate copy. So this left side is identical to this right side, and they're joined together at the center. We still consider this whole structure one chromosome, even though that there's two copies. This structure, even though it's only one copy, we still only call one chromosome. Okay, this is one unduplicated copy, this is one duplicated copy. Okay, it's both considered one chromosome, just that one has two copies to form this X shape. Okay, so that whole thing, that's my chromosome. If I want to refer to the left or right half, I use the word sister chromatids, okay? So we had chromatin. Chromatin was the loose spaghetti state, okay? Just loosely coiled. You have a chromosome, which is when it's tightly coiled, and traditionally you'll see it as this X structure. And now we have chromatid, and the chromatid is the left or right half of a duplicated copy, and they're both identical, okay? They're both the same. Go back for you. If you guys want to put that on pause to get this into your notes real quick, okay? Okay, and the next one for this is the centromere. Where do you find the centromere? In the center. And what is it doing? It's where each sister chromatid is connected, and they're connected at the centromere. Okay? Okay, so that does it for DNA, chromosomes, and all that good stuff. Now let's go into the cell cycle. So this is a graph or a chart that I want you folks to become familiar with, and I'm going to break this down and explain it to you. This chart is explaining the amount of time that a cell spends in each cycle of its life. Okay? So, interphase. Obviously, you could tell that interphase is the longest portion of the cell cycle. Okay? Next, we have mitosis. Mitosis is the next longest part of the cell cycle. And finally, the last part is cytokinesis. Okay? So there's three big processes of the cell cycle: interphase, mitosis, and cytokinesis. Now, each one of these have different events that occur within them, and I'm going to show you what those are right now. So interphase has three parts to it. The first part is called G1. The next part is called S, and the last part is called G2. Okay? That's all that we're going to focus on for the rest of today is G1, S, and G2. Now, the different events that take place in mitosis and cytokinesis we'll cover at a later time. Today is just interphase. Okay? And how many parts are there to interphase? There's three. G1, S, G2. Okay? Okay, so in G1, it actually stands for gap 1, but I like to think of it as growth 1. Okay, so we'll call G1 growth 1. And what happens is there's a high amount of protein production. So our cells are ramping up proteins left and right because it's actually growing. It's getting larger. Next, we go to S phase. And S phase stands for synthesis. And what does synthesis mean? Synthesis means to make. And what are we going to make? We're going to make more DNA. We're going to make a different, another copy of DNA. Okay. And lastly, G2, the cell grows even more. Okay, so we'll call that growth 2. Even though it stands for gap, we'll call it growth 2. The cell grows even more, and we make additional organelles. And I'll show you which organelle is made in just a moment. So interphase, this is the longest part of the cell cycle. And if I show you this chart, you should be able to tell me, yes, interphase. It's this whole purple area. You should be able to tell me, yes, that's the longest part. It's about 90% of the cell's life. So 90% of the cell is just going through its, its daily routines, cranking out proteins, copying DNA, it's growing, doing all of its good stuff. Those are the three parts to interphase: G1, S, and G2. During this whole process, the DNA is in its chromatin form. So if you were to look under a microscope and you were to see a cell, and you were to look for a cell that's in interphase, look for the cells that have loosely coiled DNA in that spaghetti state. Okay, and that should tell you that the cell is in interphase because it's loosely condensed into chromatin. Okay, it's not the chromosomes yet. There's no X structures. There's none of these yet. Okay. Okay, so here's a little animation. We have our cell. We have the different strands of DNA in the nucleus, and we have a centrosome. So G1, the cell grows. High protein production. It's getting bigger. Next we have S phase, or synthesis, and the DNA is replicated. If you noticed, I added double the DNA. Okay, so all the DNA is replicated. And now we go to G2. The cell grows even more, and we get another centrosome. Another centrosome is replicated. Okay. And that is it for today, and I will see you guys tomorrow. Have a good one.